If this is your first time here, this isn't a standalone video. It's one chapter in a complete story, the entire history of evolution, from the first replicating molecules to you. Every organism we mention is your direct ancestor, not a cousin species, not something similar, your actual lineage, an unbroken chain connecting you to the origin of life. That's what makes this different from every other evolution content out there. Most of it is scattered fragments, dinosaurs here, early humans there, the Cambrian explosion somewhere else, all disconnected. But they're not disconnected. They're all chapters in the same story, your story, and you can't understand any link without seeing what came before it. Lungs don't make sense without swim bladders. Mammals don't make sense without reptiles. Each chapter builds on the last, and nothing appears out of nowhere. 4 billion years of your deep history told as one continuous thread. If that sounds interesting, Chapter 1 is in the top right corner. I'd recommend starting there. If you've been watching from the beginning, you already know what this is. Let's keep going. This chapter describes the single most important event in our lineage's history. Every complex organism alive today, every animal, every fungus, every plant, every protist, descends from one extraordinary merger that happened roughly 2 billion years ago. An archaeal cell engulfed an aerobic bacterium, and instead of digesting it, the two established a permanent partnership. The bacterial cell became the mitochondrion, the energy-producing structure found in virtually all eukaryotic cells, including ours. This event, called eukaryogenesis, transformed a simple archaeal cell into something radically different, capable of complexity impossible for any prokaryote. The exact circumstances of this merger remain debated, but the broad outline is clear. An archaeal cell, likely an Asgard archaeon living in sediment or near hydrothermal environments, encountered an aerobic bacterium, probably an alpha proteobacterium capable of using oxygen to extract energy efficiently. The archaeal cell somehow took the bacterium inside. This might have happened through direct engulfment if the archaeon already had primitive abilities to bend and manipulate its membrane. Or it might have started as a different relationship. Perhaps the bacterium was initially a parasite, invading the archaeal cell, or perhaps the archaeon was feeding on bacterial waste products and the bacterium took up residence nearby, gradually getting closer until it ended up inside. However it began, the crucial point is that the bacterium survived inside the archaeal cell and kept functioning. It continued performing aerobic respiration, using oxygen to break down organic molecules and generating ATP. The ATP it produced leaked out and became available to the host cell. This gave the host archaeon access to aerobic metabolism without having to evolve all the complex machinery itself. The host provided the bacterium with a protected environment and a steady supply of organic molecules to metabolize. Both benefited enough that the partnership persisted. At first, the relationship was probably loose and unstable. The bacterium retained all of its genes and could theoretically survive independently if separated from the host. But over time, through millions of years, the partnership deepened. Some bacterial genes transferred to the archaeal cell's chromosome. This happens occasionally through horizontal gene transfer and provides a backup copy if the bacterium's own chromosome degrades. Once a bacterial gene existed in the host genome, the host could produce that protein and import it into the bacterium. The bacterium no longer needed its own copy of the gene, so mutations could disable it without consequence. This process repeated hundreds of times. One by one, bacterial genes transferred to the host's genome were expressed by the host, and their protein products were imported back into the bacterium. The bacterium's genome shrank as genes became redundant. Modern mitochondria retain only a tiny fraction of their ancestral bacterial genes. Humans have just 37 genes left in mitochondrial DNA, encoding only the most essential proteins that must be produced locally. Everything else the mitochondria needs, roughly 1,500 different proteins, is encoded in the nucleus, produced in the cell's main compartment, and imported into the mitochondrion. This gene transfer locked the partnership permanently. The bacterium could no longer survive independently because it lacked most of the genes needed for a free living existence. The host could no longer survive without the bacterium because it depended on mitochondrial ATP production. They had become a single organism, a eukaryotic cell, with two genomes working in coordination. The energy advantage this provided was transformative. Aerobic respiration extracts roughly 15 times more ATP from the same organic molecule than anaerobic fermentation. A eukaryotic cell with mitochondria had access to vastly more energy per unit of food than any prokaryote relying on fermentation or on the less efficient anaerobic metabolic pathways. This surplus energy enabled things impossible for prokaryotes. The first major consequence was size. Prokaryotic cells are limited to a few micrometers in diameter because they generate energy at their membranes. As a cell grows, its volume increases faster than its surface area. A larger cell has proportionally less membrane surface relative to its interior volume, which limits how much energy it can produce per unit of cellular material. This creates a hard size ceiling. Prokaryotes can't grow much larger than a few micrometers without starving their interiors of energy. Eukaryotic cells bypass this constraint by internalizing energy production. Mitochondria are internal structures, each with their own membranes generating ATP. A eukaryotic cell can pack its interior with mitochondria, scaling energy production with volume instead of being limited by surface area. This allows eukaryotic cells to grow much larger, 10 to 100 times the diameter of typical prokaryotes, without hitting energy constraints. Larger size enabled internal complexity. A bigger cell has more internal volume to organize into specialized regions. Early eukaryotes developed internal membrane systems, folds and compartments separating different functions spatially. One region might specialize in protein synthesis, another in breaking down molecules, another in storing materials. This compartmentalization improved efficiency because specialized regions could maintain different chemical conditions optimal for different processes. The nucleus evolved as part of this compartmentalization. In prokaryotes, DNA floats in the cell interior, and ribosomes start translating RNA into protein while the RNA is still being transcribed from DNA. Everything happens simultaneously in the same space. In eukaryotes, DNA became enclosed in a membrane-bound nucleus. Transcription happens inside the nucleus, producing RNA. The RNA then travels out through pores in the nuclear membrane to reach ribosomes in the cytoplasm, where translation into protein occurs. This separation between transcription and translation opened new possibilities for regulation. Inside the nucleus, RNA can be modified before it leaves. Sections can be cut out and the remaining pieces spliced together in different combinations, producing multiple different proteins from a single gene.